Okay, so now we're going to talk about chapter 17, which the main topic of chapter 17, of course, is gene expression. There's another word for gene expression. It's what we call the central dogma. The central dogma. Now I'm going to initially talk about the difference between eukaryotic gene expression, prokaryotic gene expression, and then we're going to get into the detail of gene expression. Eukaryotic gene expression. Is basically DNA being copied into what's called pre-mRNA. And that process is called transcription. Okay? Then the pre-mRNA is going to be processed into mRNA. And that's called RNA processing. Then the mRNA is going to be translated into protein, and that is called translation. You heard some of this stuff before, but it was just introductory. We're going to get into it today. The little m stands for the word messenger, because it has the message to make proteins. So, for example, let's say we're trying to make catalase. So we're going to start with the catalase gene. The catalase gene will make the pre-mRNA, which will eventually be processed to make the mRNA for catalase, will eventually make the catalase protein. That's the protein that's going to do the work for you. What does catalase do? Remember, it takes hydrogen peroxide and breaks it down into water and oxygen. And if we remember that from the lab, that's a gas. So it's not like we're copying the entire chromosome. No, if you want to make catalase, then you're going to only copy the catalase gene. That's it. You don't need to copy another gene just to make this one. All right, so how is it different from prokaryotes like bacteria and archaea? It just goes from DNA to mRNA to protein. This is again translation, and this is transcription. What's missing? The RNA processing, yeah. In DNA, in prokaryotes, you don't have pre-mRNA, so you don't have processing. That's one difference. Another difference is cellular locations, where these things happen. This one here, all the way to the mRNA here, happens in the nucleus. In eukaryotic cells, translation happens in the cytoplasm. In eukaryotic cells. So in eukaryotic cells, transcription and RNA processing happens in the nucleus. Translation happens in the cytoplasm. In prokaryotes, the whole thing happens in the cytoplasm. Why not the nucleus? Because they don't have a nucleus, that's why. They're prokaryotic cells. They don't have a nucleus. So it's only in one location. Are we okay so far? Picture before I erase. So, put together, that is called gene expression, that is called the central dogma. That's basically the function of your DNA. Your DNA has the instructions, or is the instructional manual to making your proteins. And do you remember what we said about proteins? Proteins do everything, yeah? So these are the guys that are actually working for you, but the information to make them is in your DNA. Everything about you is written in your DNA. Okay, so now that we have a bird's eye view of the event here, we're going to now start talking about each event in more detail. Know that whatever I say about transcription here, for the most part, it's the same as here. Same thing with translation, for the most part. Of course, there are differences, but you'll learn those more in microbiology, because microbiology focuses on bacteria. So we're going to focus more on the transcription, processing, and the translation. All right, let's go through it. Now that you're all stretched out, let's do it. Good. I know. Long day today, you guys. But bear with me, please. You'll thank me for going over it when you get home. All right, listen. We're going to do the first step. The first step is transcription. Here we go. Transcription is DNA to what? RNA. Good. So I'm going to specifically talk about the eukaryotic one. DNA actually going to pre-mRNA. So this would be called the primary transcript, the initial transcript. It is not mRNA yet. Once it becomes, it's not mature mRNA yet. Once it processes, it becomes mature RNA. We'll talk about that. So the first step is transcription. That's DNA being copied into pre-mRNA. Now, we realize something. When we look at genes, we realize that, yes, here's a gene for catalase, for example. But ahead of this gene, there's a control element called the promoter. That's like the light switch by the door. When I walked into this room, how did I turn the light on? You guys sat in the dark, but I turned the light on by pressing the light switch. How do you turn this gene on? Here's the switch for it, the promoter. The promoter has an enzyme that comes and binds to it to initiate transcription. This enzyme will not bind to the promoter until it's been invited to do so. Oh, okay. So this is like the popular kid on the block. The popular kid on the block, his name is RNA polymerase. You're throwing a party and you want to invite the popular kid. He has to be invited by a certain person. And that person has to be invited by another person. And so forth and so on. So for you to have a party, you have to invite Jack to the party. When Jack is there, he invites Sue to the party. Once Sue shows up, she invites Mark to the party. Whatever. Just throwing names. All right, once everyone is there, here's Bill. Bill invites RNA polymerase, the popular guy. So if Bill is not at the party, he's not coming. You see that? And if Mark is not there, then Ben is not coming. You can work backwards on it. These guys are called transcription. Obviously, they're proteins. Factors. There are hundreds of them. You can get a PhD of just this alone, right there. If you want to study that. It's amazing. Hundreds of them. And they have to come in at a specific order. That's what I'm trying to give you an analogy of. I can't remember the names anymore, but let's say Jack before Sue, Sue before... Eventually, the whole goal is to invite the popular kid, RNA polymerase. Once everyone is at the party, he's so cool. He waits for everyone to be there. Then he shows up. Who is he? He is RNA polymerase. And what does he do? He transcribes the gene, the sequence, into pre-mRNA. That's what he does. He copies your DNA into mRNA. Now, before I show you what the end result of that is, I have to get you to realize something about things that you do on a daily basis. You may not know this, but you'll probably know it now and realize this, that everything about you has 3 events: initiation, elongation, termination. You're not going to understand this right now, but you will soon, in the next minute or so. I'll give you an example. When you came in here today, here's your initiation event. You woke up, took a shower, got dressed, looked for your keys, found your keys, put your keys in your ignition, and you're about to start your car. All of that is initiation of driving. Are we catching that? Once you start your car is running. That's elongation of driving. You pull out of your driveway, you go down the street, you turn left, you turn right, you get on the highway, you come into this parking lot, and you park your car. All of that is elongation. Once you get into the parking spot, what are you going to do? You're going to turn off your engine. That's termination. Every event in your life has those 3 components to it, including your life itself. You were conceived, you developed in your mom's womb, and then you were born. That's initiation of life. Now, that's debatable when life initiates. Let's assume it initiates at birth time, at birth. Let's say that. That's initiation of your life. Then you're living, you're growing, you're causing problems. You're graduating high school, graduating college, you're becoming whatever. That's elongation. And after a long life to you and I, what's the 3rd step? Termination. Everything about you is like that. Why not transcription? So there's a transcription initiation event. Initiation. There's a transcription. Elongation event. And there's a transcription. What was the 3rd one? Termination. So, when Bill invited Sue, Sue invited Mark, and Mark invited whatever, and then they all invited RNA polymerase. That's this one. That's this one. That's the initiation part. That's you taking a shower, getting dressed, finding your keys, going to your car, putting the keys in, and you're about to start. Once the RNA polymerase binds to the promoter and it's about to start, that's now this one. Once it starts, it's this. Once it actually starts making the RNA, now it's this, elongation. Eventually, the RNA polymerase, which is the only enzyme involved in transcription, thank God. You remember in DNA replication? There was a whole list. Topoisomerase, then the other topoisomerase, then the helicase, then the single-stranded binding protein, then the good primase, right? Then the DNA polymerase 3, then DNA polymerase 1, and the last one was DNA ligase. That's DNA replication. Thank God. This guy, it's only one enzyme. RNA polymerase. That's it. So, RNA polymerase will sit on your DNA and start making the RNA. That's this one, elongation. Eventually, it's going to get hit a sequence on your DNA called the terminator. Like Arnold, right? Arnold Schwarzenegger. The terminator sequence. And it will tell RNA polymerase, you're done transcription. Get off, you're done. But by then, you'll have your pre-mRNA made. By the time the RNA polymerase reaches the terminator sequence, which is a sequence at the end of your gene, the RNA is already been made in the form of pre-mRNA. That's transcription in eukaryotic cells. Then we're going to go to the next event. The next event is RNA processing. RNA processing will take the pre-mRNA and process it into what? Yeah, into mRNA. So RNA processing will take your pre-RNA and will process it into the mature mRNA. The word mature is not this one. This, remember, means messenger. Mature means processed RNA. When I say mature, I mean it's been processed. So this is the mature mRNA. This is the primary transcript, the pre-mRNA. Okay, what are the processing events? It's sort of like a chocolate factory. You make chocolate, the processing events would include wrapping the chocolate, putting it in the box, whatever, transport, and that's the processing event. Okay, what's the processing events here? It starts out with something called 5' capping. Please follow me here. This one is, each event has a function, and I'm going to explain to you what they are, and I'm going to expect you to know it. The 5' capping is basically this. I take the pre-mRNA and I put a cap on it at the 5' end. It's a special guanine. It's a special G. It's capping. It's basically saying, hey, that's the 5' end. That means the other one is the what end? Good. Watch this. Who is that? Who is it telling that to? Hey, that's the 5' end. It's telling it to the ribosome, the translator. It's saying, hey, ribosome, I know you're about to translate this. This is where you start. Don't start here. I want you to start here. So the 5' cap is responsible for ribosome binding for translation. Because the ribosome is your translator, and we talked about that before. Okay, what happens to mRNA that doesn't have the cap? It doesn't get translated. mRNA that does not have the cap, the ribosome goes, I don't know which end is which. Forget it. I'm not going to touch it. You must cap it to translate it. Because the cap tells the ribosome, come and bind over here and go this way. Without the cap, it does not get translated. Now watch, in bacteria, it doesn't need that. Because bacteria, remember, does not have RNA processing. So what we're doing here is exclusively eukaryotic. All right, that's similar to me saying to you, put your cap on before you leave the house. Because that's the only way I can identify you. If you don't have the cap on, I don't know who you are. So capping is important for ribosome binding. Okay, next event. The next event is this. When we look at our genes, our DNA has exons and introns. Exons and intron regions. That's on your gene. For example, this is the catalase gene. Listen to this, please. This is the catalase gene on your DNA. For example, the gene is made up of 3 exons and 2 introns. To make the catalase, you need the exons, not the introns. So watch what happens here. This is your DNA. It says to make catalase, you have to remove the introns. But don't you dare remove them on the DNA. You don't want anything to happen to your DNA, trust me. Here's what I'm going to do. I'll make a direct copy of the gene in RNA form. So this one will look like this. Exon, intron, exon, intron, exon. You can do whatever you want with this one, but don't touch my DNA. The first time I do this to you is the last time I'll do this to you, because you'll be dead. See what I'm talking about? I don't touch my DNA. I like it the way it is. All right, here's what I'm going to do. I'm gonna give you a direct copy of my gene in RNA form. We call it the pre-mRNA. You can do whatever you want with this one. Don't touch this one. So what I'm going to do with that is initially cap it so that the ribosome can bind there when the time comes. Second event, I will remove the introns because I don't need them. Catalase is the 3 exons coming together. That means I have to splice out the introns, and that's the next event. Splicing out of introns. So it's going to look like this at the end of it. It's going to look like this. I'm going to have the cap, because that's already done. And now all I'm going to have are the exons cut and pasted together. What happened to the introns? They were spliced out because I don't need them. They are intervening sequences. The introns will not make catalase. It's the exons that make the catalase in my example here. So it's a cut and paste. You remove the introns and you paste the exons. I'm just assuming it has 3 exons. I don't know how many exons it has. But I'm not done yet. The third event is adding what is called a poly-A tail. Poly-A tail. Adenine. A stretch of adenine. And once I'm done with it, it's going to look like a tail. But where do I add it? On this end. So it's going to look like this. Watch this. At the end of it, it's going to look like this. A. How many hundreds of them? Maybe a couple of hundred. I don't know. Who cares? So it's called the 3' poly-A tail because it's added to the 3' end. That's your mRNA. That's the one that's leaving the nucleus. That's the processed one. How was it processed? It's been processed by capping, by splicing, and the poly-A addition. Let's review the function because I did not tell you this one. The function of the cap is ribosome binding. An mRNA without the cap does not get translated into protein. The removal of the introns. Why? Because they're intervening sequences. What would happen if I forget an intron inside here? I will not get catalase. What will I get? I don't know, but it's not catalase. To get catalase, I have to have the exons, no introns. If a mistake happened and I left this intron behind, I know for a fact I'm not getting catalase, but I won't be able to tell you what protein I'll get because it won't make sense anyways. The cell says, I don't know, what is that? I don't know, nobody knows. I wanted to make catalase, now I'm making this weird thing. I don't know what that is, and good luck with that. First of all, you won't have the catalase. Second of all, you're gonna have a protein that you have no idea what that is. You're gonna suffer twice. You don't have the protein that you want, and now you have something that you don't know what it is. You got problems now. So the best thing to do is remove the introns. Now, I'm ready to make catalase. Now watch this. You add the poly-A tail for another function. The poly-A tail allows the mRNA to leave or exit the nucleus. What happens if I don't add the poly-A tail? The mRNA will never leave the nucleus. That means it will not be translated. Why? Because the ribosome is in the cytoplasm. If you remember from chapter 6. Where is the ribosome? It's free-floating in the cytoplasm, and it's attached to the rough ER. Those are where the ribosome locations are. I know there's some in the mitochondria. We're not going to talk about those with this one. This is the mature mRNA. It's been processed. It has the cap. The introns have been spliced. It has the poly-A tail added for exiting the nucleus. Now watch this. This area here is going to be called the 5' untranslated region, UTR. Untranslated region. That means it's a region on the RNA that is not translated by the ribosome. Just like it says. And then there's another one like that on this end. And that's called the 3' UTR. They're both called untranslated region, but it also tells you their location. One at the 5' end and one at the 3' end. They have their jobs. The 5' UTR region regulates translation. It controls, should we make the protein or not? So it's involved in regulating protein translation. Listen to the function of the 3' UTR. The 3' UTR stabilizes the message. Imagine this scenario. Watch this. Watch the show, please. I go through all the trouble of making the RNA. I cap it, I splice it, I add the poly A tail. And as soon as I make it, it breaks down. Then I can't make the protein. Do you agree? Mm-hmm. If as soon as I make it, it breaks down, the ribosome says, okay, I'm about to bind to you, and you'll break down on me, then I can't make the protein. So that's the function of the 3' UTR. It stabilizes the message. So that when I do make it, I'm stabilizing it. Its half-life is long enough for the ribosome to bind and translate, another one to bind and translate, another one to bind and translate. Now, do we have enough protein? Yeah, now it breaks down. Only when it realizes we've made enough protein, I don't need this anymore. It's fascinating. Everything has its job. And if you mutate one of them, it's at the consequence of that job, whatever that is. If you mutate the 5' UTR, you're not going to have translation regulation. If you mutate the 3' UTR, it's going to affect the stability. If you don't add the poly A tail, it's stuck in the nucleus. It will never get translated. If you don't put the cap on, the ribosome will never bind to initiate anything. It's amazing how everything has its job. So that's transcription, that's RNA processing. What's the next step? Translation. Here comes the fun one. Bear with me, please. Translation is the next event. Now we have the message. It's a mature message. It has the cap. It's been spliced. It has the poly A tail. Everything is good. I am ready to go. And what are we doing? We're doing translation. We're getting the ribosome involved in this now, because that's the translator, the ribosome. So now, in this case, I'm going to write a sequence out. So the sequence, let's say, for example, the sequence is CCG, GCU, AUG. Why U? Because what am I working with? RNA. There are no T's. AUG, GGG, CAC, UGC, UGA. That's my sequence. This is 5'. This is 3'. By the way, when we go back here, one of the things that I want to show you here, when we transcribe something, we're making DNA to RNA. So if this is AGCT, then your RNA is going to be UCGA, because when you have an A on the DNA, it's going to be a U on the mRNA. And what normally happens is this. Let me show you something here that I forgot to mention. Let's say we have the DNA sequence. and it is like this, A, G, C, T, G, C, A, for example. The double-stranded DNA is gonna be T, C, G, A, C, G, T. That's your DNA. Am I right? This is 5 prime. This is 3 prime. This is 5 prime, sorry, 3 prime, thank you. This is 5 prime. We talked about this before. The question is, which one of the 2 strands will RNA polymerase, this guy, the party animal guy, gonna copy? It's always gonna be this one, because that one is called what is called the template strand. It also goes by another name called antisense. It also goes by another name called, I mean, how many names do you have to go by? But this one has 3 on them, non-coding. So the template strand is the same as the antisense, which is the same as the non-coding. So when you go and transcribe, which what we talked about, to get this transcript that I wrote up here, you have to copy the template strand. Why? Because RNA transcription is also 5 prime to 3 prime. I told you before, all nucleic acid synthesis is 5 prime to 3 prime, regardless if we're making DNA or RNA. Stay awake with me, please. So for me to make the RNA 5 prime to 3 prime, I have to read this strand, 3 prime to 5 prime. So I have to read this one. And now I'm gonna do this. For every T, there's a, and because this is 3 prime, this is good. For every C, there's a, for every G, there's a, for every A, there's a U. Watch out for that. For every C, there's a, then what's the next one? Okay, then what's the next one? Good. And a little 3 prime. So I added that in there. So after transcribing the template strand into RNA, let's say I got this sequence. Now I'm about to make it or translate it into protein. Forgot to say that earlier on, but now I said it. I had to put it on camera too for you guys.
Does it always have to be in the order of 3 prime to...
Yes. It's gonna read 3 prime to 5 prime because it's gonna have to make the RNA 5 prime to 3 prime. For it to make the RNA 5 prime to 3 prime, it has to read the template 3 prime to 5 prime. It's no different than we talked about when we said the leading strand and the lagging strand. So now that we have this sequence has been capped, been spliced, and has the poly A tail. It left the nucleus, and the ribosome is about to bind to it. Here we go. How many subunits does the ribosome have? 2, good. I don't want to show it in the camera there. All right, 2. There are 2 subunits, a large subunit and a small subunit. Look what happens. It's always going to be the small subunit that comes in play first. The small subunit comes in play first. Why does it bind here, not there? Because what's on this end? The cap. Very good. That's why the cap, the special G, that's why the cap is important. The cap is saying, hey, I'm the start, I'm 5 prime is right here. Bind here, don't go over there. If you go over there and go that way, you're gonna produce, you're not gonna give me catalase, you're gonna give me something else. I want catalase, it goes that way, for example. So the small subunit of the ribosome, small ribosome subunit is the one that comes in play first. It binds to the capped end of the mRNA. And look what it does. Listen to how I'm reading the sequence, because that's how the small subunit will read the sequence. C, G, G, C, U, AUG, GGG, CAC. Did you catch that?
It's sped up at one point. Yeah, and didn't speed up. It started reading it as groups of 3. That's what I was doing. I was saying it one at a time. C, C, G, G, C, U. But once I get to this AUG, I recognize, oh, that's the start codon. I am now going to read it 3 nucleotides at a time. I'm going to say it this way now. AUG, GGG, CAC, UGC, UGA. I read it in triplicates. Once I recognize or identify the first AUG, I read it in triplicates now. And the triplicate is called a codon. So what the small subunit is doing is scanning for that first AUG. And how does it do it? It reads it one at a time first. But once it finds the first AUG, it says, I got it, because that's the start codon. So now the small subunit is sitting right here. It is no longer here. It is now here. And it will now read the mRNA in triplicates called codons. So, there are 64 genetic codons. There are 64 codons. Here they are. You do not memorize these, please. You will have this on the exam to reference, but you just need to know how to use it. And we're going to use it together before we leave here today. There are 64 codons. 61 of them code for amino acids. 61 of them. Look at it. There are 64 total. Only 3 say stop what you're doing. Stop translations. They're called stop codons. So UAA is a stop codon. UAG is a stop codon. And UGA is a stop codon. All the other ones code for amino acids. Now I'm going to have to bring the screen back up so I can finish the diagram. I'm going to look back here. I have AUG as the first one. Which amino acid does AUG code for? MAT. So I'm going to just write it here because I'm going to have to get rid of that screen. Okay, the next one is GGG. It's a codon. It's made up of 3 nucleotides. Position 1, position 2, position 3. Position 1 is G. Position 2 is G. Position 3 is G. So GGG gives me glycine. That's how you read the codons. Let's do it again. You guys tell me. What does CAC give you? Let's do it. C is the first position. A is the second position. C is the third position.
Histidine.
It's histidine.
Histidine.
Yeah, histidine is something else. Yeah.
It's
Histidine. Yeah. UGC. What does that give you?
UGC.
Yeah. Cis. It's called cysteine. Very good. You don't need to memorize the amino acids at all. You will have access to this on all the exams from this point on. And the last one is UGA. What does that give you?
Stop.
That's a stop codon. It's telling the ribosome to stop what you're doing. And what's the ribosome doing? Translation. Now, before I bring the screen back up, there are some characteristics of the genetic code that you must know. And this is a little bit involved, at least initially, then hopefully you'll get it. Listen carefully, please. There are multiple ways of getting the same amino acid, as you see here. There are 4 ways of getting threonine, for example. ACU, ACC, ACA, ACG. Why? Because the third position of the codon is called the wobble position. W-O-B-B-L-E. Wobble position. I also refer to it, to remember it, the whatever position. Get it? Whatever, wobble, whatever, WW. Because AC whatever gives you threonine. AC whatever gives you the same amino acid. So we say that the genetic code is therefore redundant. because of this. Multiple codons give you the same amino acid because the genetic code is redundant. See, if you come in here with 5 cell phones, I will go to you and say, you are redundant when it comes to your cell phone. That means you have multiple cell phones. Multiple codons code for the same amino acid. It's because the genetic code is redundant. But why is the genetic code redundant? Because the third position is a wobble position, that's why. Not all the time, but most of the time. Now watch this. Is there a wobble position for AUG? No. There's only one way to get methionine, and that's AUG. That's it. Do you see any other methionine? No. So when it comes to mutations, because genetic mutations happen, right? The random, rare, DNA polymerase makes a mistake and overlooks it. Mismatch repair doesn't fix it. Excision repair doesn't fix it. Now you have a mutation. If you have to have a mutation, ask for one right here. Do you see that? Because if you're ACC and I am ACG, we're different at the DNA level, but our protein still is the same. And we call that a silent mutation. You'll see that later. But you don't want a mutation here. Because if you mutate AUG to AUA, you're going to go from methionine to isoleucine. This does not have a wobble position. The wobble position is when you have AC, whatever gives you the same amino acid. And because of the wobble position, the genetic code is said to be redundant. Look at leucine. There are 6 ways of getting leucine. A lot of students understand that part. And then they go to this one and say, oh, they think I'm contradicting myself. I am not. When I talk about this one, I am not contradicting what I just said. Let me repeat redundancy again. You can tell it's a problem for students. Redundancy is multiple ways give you the same amino acid. But if I ask Kai, and I will, Kai, what does UUU give you? UUU. Look on the screen, sir. What does it give you? Okay, does it give you anything else?
No.
Good, because it's not ambiguous. The genetic code is not ambiguous. UUU gives you phenylalanine, and that's it. You know, if Kai said, well, sometimes phenylalanine, sometimes something else, oh, that we're in trouble. We are in trouble. Imagine your cell says, I'll sometimes make catalase for you when you need it, but I sometimes won't. Think about that. Your cell says, I'll sometimes make catalase when you need it, but sometimes I won't. That means sometimes you'll be alive, but sometimes you'll be dead. And I'm worried about the part where I'm dead, not alive, you see? And you can't do that. You can't say, I will sometimes have life, but sometimes I won't. Then life becomes a chance kind of thing. Oh, no. When I have UUU coding for phenylalanine, that means I need phenylalanine. That's what I'm coding for. When I have GUU, I need valine. When I have GAU, I need aspartic acid. You see? I don't have this, I cannot afford this ambiguity where sometimes I'll get alanine, but sometimes I won't. Are you kidding me? That means sometimes I'll make catalase and break down the hydrogen peroxide so it doesn't hurt me, but sometimes I won't. That means the hydrogen peroxide will hurt me. Then life does not exist. You can't have this. You can't have ambiguity and have life. The reason, at least part of the reason why we exist, is because the genetic code is not ambiguous. UUU will code for phenylalanine in my kidney, in my liver, in my heart, in my brain, if I'm human, if I'm a plant, if I'm a protozoa or bacteria, because the genetic code is not ambiguous. And I can't stress that enough here, because a lot of students make a mistake between redundancy and the genetic code being not ambiguous. There's a difference, and I'll say it one more time. Redundancy means multiple ways gives you the same amino acid. Not ambiguous means when you have this codon, it only gives you this amino acid. That's it. ACU, threonine, that's it. GCU, alanine, that's it. So it allows us to do what we're about to do. Predict the protein sequence from a mRNA sequence. Because of the not ambiguous, we can do that now. We can predict the protein sequence from an mRNA sequence. So I have this sequence here, and I'm about to make a prediction of what the protein sequence is going to be like. Obviously, you can tell what it is on the screen over there, but I copied it because I had to bring the screen up. So watch, please listen to me. You need to listen to every event here. The small subunit will scan for the first AUG, and that's the start codon. The first AUG serves two functions. Number one, it's the start codon. Number two, it brings in the first amino acid. And the amazing thing is, how does it do that? How does it know that AUG codes for methionine? It doesn't know that. It knows this. There are tRNAs out there in your cytoplasm. And a tRNA is loaded with an amino acid. That's what the tRNA, T, is transfer RNA. It's like they're delivery trucks. Imagine every delivery truck has a license plate. It's looking for the license plate that is complementary to AUG. It's looking for the anticodon. This is the codon on the mRNA. What is complementary to the codon? The anticodon on the tRNA. So for every A, there's a, not T, it's RNA, U. For every U, there's a, for every G, there's a, there we go. So what it's looking for is, among all the delivery trucks, who has the UAC license plate? That truck is always going to be loaded with methionine. That's how it does it. The anticodon, UAC, will always come with methionine. And then the large subunit is invited in. The large subunit has 3 rooms: the E room, the P room, and the A room. E is for exit. P is for peptidyl. Your slides talk about the names. And A stands for aminoacyl. There are like 3 chambers in the large subunit. So the large subunit comes with 3 chambers, and it sneaks in from behind, and it orients itself so that the P site is where this tRNA is. So this first tRNA starts in the P site. The E site is empty, and the A site is empty. So look what this is. The small subunit bound here. Looked for the AUG. Got it. Invited the first tRNA that has methionine. Then invited the large subunit and made sure that the first tRNA is positioned in the P site. That is translation initiation. That is the initiation of translation. Initiation. Good. The next event is going to start translation what? Elongation. That's when the ribosome reads this GGG, the codon. The next codon is going to be read by the ribosome, GGG. And it says, hey you, tRNA, with the license plate, what? What's complementary to GGG? CCC, come over here. And this tRNA will be carrying glycine. By the way, when you go use that table that I just showed you, you're always using the codons. Do not use the anticodons. I'm just showing you what's happening. By the way, there's hydrogen bonding between these. There's hydrogen bonding. That's how they're held together. Complementary base pair. There's hydrogen bonding there. But look where it came into. It came into the A site. now you're in elongation. Once it starts reading the next codon, now you're in elongation. Elongation, yeah. Translation elongation. Okay, now look what happens now. This amino acid breaks off of this and gets transferred here. So this methionine will now be sitting here, leaving this tRNA empty. The truck got unloaded. So this tRNA is now empty. This tRNA has now glycine and methionine. This one is in the A site, this is in the P site, and the E site is still empty. The next event is that the whole ribosome moves over one codon. So whatever that was in the E site is now, whatever that was in the P site is now in the E site. Whatever was in the A site is now in the P site, and the A site is empty again. Let me show that to you. Here's the P site, here's the A site. This is in the P site. Now, when it moves over one codon, this will now be in the E site. The empty tRNA is now in the E site of the large ribosome. This one will now be in the P site, and it will look like this. And the A site will be empty again. Now, underneath the A site is the CAC codon. Underneath the P site is this codon. Am I right? Hang on, let me see. Yep. And underneath this one is this one. This guy will leave empty. The empty one will leave. It will leave and reload somewhere else with methionine. See, the E site will now empty. The empty tRNA will leave empty and go reload somewhere else. So it can come back and deliver methionine if you want it to. The P site will now have the glycine and the methionine. The A site is empty. It's going to read this one, CAC, because we're still in elongation. What's CAC going to bring in? Histidine. Very good. But what's the anticodon? C. What's complementary to C?
G.
What's complementary to A? What's complementary to C? Good. How are these binding? Hydrogen binding. You see that? And it's going to repeat again. Now this time, both of these are coming off. And they're going over here. Histidine, glycine, methionine. The whole thing is going to shift over one. And whatever was in the P site is now going to be in the E site, and it will leave and reload. Whatever that was in the A site will now be in the P site, leaving the A site empty to read this one. What's going to happen is cysteine is coming in. So now you're going to have this at the P site. You're going to have GUG, which is this one, because this one will have left already. And you're going to have the histidine, the glycine, and the methionine. The E site is going to be empty, the A site will be empty. Now it's going to read this one. UGC is here, now CAC is here, and GGG is here. Oh boy. GGG is here. You gotta watch a video on this. Now, the next tRNA comes in loaded with cysteine. Okay, so the next tRNA will come in, and that's gonna be A, C, G, anticodon, and that's gonna have the cis. what's gonna happen next? Can anyone tell me?
The M is carried over.
Just the M?
I think all three.
All three of them. All three of these are going over here. You're building the protein.
So it's constantly like a block of.
Oh yeah, it keeps growing and growing and growing. Initially you only carried one over, then two, then three, then four, then five, and six. How many are you gonna carry over? It depends on how long the protein is.
So what happens to the one that it was taken from? Like that.
Well, that's what I said. This one goes in the E site and leaves empty for reloading.
So it's gonna be reloaded one more time?
Oh yeah, it will go and reload with the cysteine to participate somewhere else.
But this is steam from where?
From your cells, from your nutrients, from your food.
So all of this is just happening inside.
Yeah, when you eat protein, when you eat chicken, you're providing the amino acids to load onto your tRNA. Next time you eat a hamburger, your body is going to take the hamburger and break it into its amino acids to start loading. The methionine goes here. The cysteine goes here. You see, it loads the trucks. So you can use them to do this. If you don't eat the protein, you're going to have empty trucks. Good luck doing that. That's why they say eat a healthy diet. Because we can make some of the amino acids. We can synthesize some of the amino acids. But there are some that you must take or get from your food. If you're not eating a healthy diet, some of the delivery trucks will be empty. And if your ribosome taps into that one next, I need this one, and brings it in empty, oh, you're in trouble. If this guy comes in empty into the A site, you can't build on it, you're in trouble. So a healthy diet saves you from that. All your tRNA delivery trucks will have all their amino acids in there, and you can start tapping into them to do this. Very good question, though.
Thank you.
You're welcome. This empty tRNA will leave through the exit site. This growing chain will go back to the P site, will go to the P site, leaving the A site empty again. Now look at the next codon that it reads. The next codon that it reads is a stop codon, UGA. What have you reached? You've reached the termination step. Once the ribosome reaches a stop codon, here's what happens. It stalls. It's looking for the tRNA that's complementary to UGA. I don't have one of those. There aren't any. So it stalls looking for it. Where is it? Where is it? Where is it? It doesn't exist. So a release factor, which is another protein, will come in, bind into the A site, right over the stop codon, and will order the whole thing to fall apart. Because by then, you're done. Termination. Let me show you that last step using the slides. It's involved, I know. You must watch an animation on it to visualize it. Wait until it warms up a little bit, you guys. Hang in there. Come on. It's probably going crazy. Why did you go up and down, up and down? There it is. Look at that last part right here. We talked about all of those things. The processing. Here's the EPA site. Here's the small subunit looking for the start codon. Here it is inviting the first tRNA with the methionine in there. Here's the codon anticodon recognition. Here are the EPA sites. Remember, the first tRNA binds to the P site, goes through the P site. The E and the A are empty, and it starts the elongation.
Wait, what's GTP?
GTP is an energy molecule, because this requires energy. It is anabolic.
I had a feeling it was.
It's similar to ATP. So here's the cycling that I just told you about. It reads the A site. It reads the codon located over the A site, brings in the tRNA. It transfers the growing chain onto the one that just came in, leaving this one empty to leave through the E site. And then the growing one and now is in the P site. And the cycle repeats over and over and over until it reaches a stop codon. Once it reaches a stop codon, it stalls. It's looking for a tRNA that is complementary to this codon, but it does not exist. So it stalls. The release factor, which is a protein, binds to that site and orders the whole thing to disassemble, because then you're done. You have reached translation termination. The protein folds and goes and does its job. The two ribosome subunits fall apart. They can reassemble and do it again somewhere else. The mRNA, if you don't need it anymore, will break down, but if you need it again, you have the gene to remake it. It's fascinating. This event is happening every second of your life in every cell in your body, and your life depends on it. So when we look at your mRNAs, we find a very clear example of your cell being extremely efficient. You really think your cell is going to do this? Here's the start codon, here's the stop codon. You really think your cell is going to bind the ribosome and wait until it finishes before it binds the next one? No. Your cell is going to do this, bind the first ribosome, and by the time it gets here, I'm binding the second one. And by the time these two gets here, I'm binding the third one. By the time these three are here, I'm binding the fourth one. For efficiency, I'm going to produce what's called a polyribosome. Look at it. If you look at the thin line right here, that's your mRNA. You can see it a little bit better right here. That's your mRNA. But these structures here, this is a real electron microscope image. These structures here are your ribosomes going to work, making proteins. They form what's called a polyribosome for efficiency purposes. Yes, sir?
What's the protein?
Say? Oh, it's coming down, but you won't see that. That's too small for that. It's coming out of the ribosome, but you won't see that. So it would be similar to what you see here schematically. But you notice something. There are ribosomes that are already completed their job. And while there are ribosomes still starting, and then some all the way throughout in different stages of the job. Because your cell is efficient. It wants to make proteins, and it wants to make it now, and at this much. It wants to make it at this level. It doesn't have time to wait for this to happen. It's going to produce what's called a polyribosome for efficiency purposes. The last topic that we're talking about, thank God, right, is genetic mutations. I hope you get this impression that if I change the sequence of the DNA, I will therefore change the mRNA. Am I right?
Yes.
Which gives me a chance that I will change, there's a chance I will change the protein. Am I right?
Yes.
Unless the change is in the wobble position. Do you see that?
Medicine.
If you change the DNA in the wobble position, then you might change the DNA, but you will not change the protein most likely. That's when I said, if you have to have a mutation, ask for one in the wobble position, because you'll be fine. Watch this. There's an example of genetic mutation here. This is your normal hemoglobin. They're only looking at one codon. Obviously, there's more codons, but they're only looking at the codon that's more commonly mutated in humans. Look at the change from T to A. That's it. By mistake, you go from T to A. I mean, you think, okay, what's the big deal? Well, look at it. T gives you A gives you U. So if you change this sequence, you're going to change the RNA. You're going to also change the protein. So we talked about in chapter 5, the structure of your protein is determined by the primary structure. The overall structure of your protein is determined by its sequence. Do you remember that? Well, you change the sequence. So you get what I call a ripple effect. If you change the primary sequence, you're going to change the secondary sequence, which means you're going to change the tertiary sequence. And this one is hemoglobin. It has quaternary structure. So you're going to change the quaternary structure. You're going to change the shape of the red blood cell. You're going to change its
Function.
There you go. And you said it loud, nice. Function. No wonder why it's devastating. So this is an example of a single base change causing a drastic effect. So that single base change is called a nucleotide pair substitution. Instead of A, you by mistake put T. Now, what's the consequence of that? It depends on where it happened in the protein and the gene, and where it happened in the codon, and which mutation. But here are the possible outcomes. One possible outcome of a nucleotide pair substitution is nothing happens to the protein. Silent mutation. You are ACC, I am ACG. So we're different at the DNA level, but the protein works exactly the same. That's a good one because, we dodged one here. Yes, we're different at the DNA level. So if you leave your DNA behind, I'll be able to figure out who you are. But at least functioning, we're fine. We both have the same protein. We're both okay. Nobody's sick here. This is different than this one. A missense mutation is what I just showed you. That sickle cell anemia is a missense mutation. Because what happened? You changed the sequence of the protein. That's what happened. This is a base substitution that caused a missense mutation. So the missense mutation is the effect. This one here is the cause. So the missense mutation means a genetic defect or mutation that caused a change in the amino acid sequence. This is different than a nonsense mutation. Look what a nonsense mutation will do. The normal stop codon is here, but a mutation happens here that makes it here instead. A stop codon. So watch this. Catalase is from here to here. That's catalase. You put a stop codon right here, you make it smaller. That's not catalase. This is part of catalase, but it's not catalase. This one breaks down your hydrogen peroxide. This one does nothing. Then you suffer the consequence of the hydrogen peroxide because catalase is a detoxifier. So what happens is this. A genetic mutation caused a stop codon to be early. Instead of the stop codon being here, it is now here. You're making part of catalase, but not catalase. The full catalase will detoxify, will break down the hydrogen peroxide. This one does nothing for you or to you. You're now being poisoned by the hydrogen peroxide. Yes, sir?
Wait, you've taught us multiple times about feedback inhibition or...
That's with the normal protein. That's when the protein is working fine, and it's the control mechanism is working fine. We're talking about now a protein that you have only part of. Can you drive on half of a tire, Lawrenson? Yeah, there you go. That half of a tire does nothing for you. Yeah? So it's the same thing. Part of the catalase does nothing for you. The full catalase, you're fine. So nonsense mutations is a base substitution that resulted in an early termination site, in an early stop codon site. Yes, sir. Real quick.
Okay, so for missense, it codes a different type of protein.
The sequence changes.
Okay, the sequence changes, but would that protein work for something else?
No, normally, it's still hemoglobin, but it's a defective hemoglobin. It's sort of like having a small leak in your tire. Is it working the same way it's now? A nice round filled tire with air is fine. It's better than something that's leaking on you. This is a protein that has changes in its structure. Well, now the problem is, how much function do you lose out of it? Sometimes a minor change in structure abolishes the function completely. Sometimes it changes the function a little bit. It all depends. So Kai, come on up here while you're talking. Come up here. I'm gonna use you as a demo for the next one, but you cannot be on the camera there. So stand over here, look at them, put up some codons for me, like this. There you go. So this is called the reading frame. If I am a ribosome, I'm gonna read this codon, this codon, this codon, and that codon. He's sketching on really good there. Good. So let's say I'm making glycine, alanine, arginine, aspartic acid. That's the codon. That's the reading frame. Watch this now. What happens if DNA polymerase 3 decides to do this? I'm not gonna copy this one. That's a deletion. So I'm gonna read this codon just fine. When I go to read this one, pay attention with me, please. What do I need to do?
Read part of his.
Read how many of his? There you go. I'm going to steal one. Let me have that. Now, am I reading the same codon? No.
That's a different one.
Yeah.
That's a different
Now, what am I going to do with these two?
You're gonna take it.
Give me that. Yeah. So that's called a frame shift. So initially, this was the frame. Now, I caused a shift. A deletion caused a frame shift. Now, look at me creating another frame shift. And here's another example of a frame shift. Watch. Insertion. You see that? Deletion, frame shift. Insertion, frame shift. Watch. I can read this one fine. I can read this one fine. When I go to read this one, how many of these do I need? It's still a frame shift. When I go read this one, I need that. He caught on. There you go. Good job. Go ahead. Thank you, sir. Those are devastating. I'll show you an example of a disease that's caused by, at least partly caused by, frame shift mutation. Now, I'm not saying all the people that have this is caused by a frame shift. I say at least partly. Come on. Yeah. There needs to be more impressive images than that. Hang in there, guys. Hang in there. Google. Hang in there, you guys. I gotta tell you about what you need to do real quick before you leave. That's an example of one. And there are more gruesome pictures than that. It's basically benign tumors all over your body. I am not saying that frame shift is a cause of it. I'm saying in some cases it is. They could be caused by other. It's the largest gene in humans. So it's all these kind of defects it becomes a major target for all these kind of defects. These people are predisposed to cancer and so forth. Although they're benign, but they run a higher chance of developing cancer later in their life. They just give you, they typically don't hurt until some of them start pushing on the nerves, then they have to deal with those. Yeah. Before next Monday, so we're done with it. Before next Monday, you have to study chapters 14, 15, 16, and 17. Do you remember what's going on with lecture exam 4?
Yep.
What's going on with that?